Hello, I'm Dr. Tim Graubert, the director of the Hematologic Malignancy Program at the Mass General Cancer Center. I'm joined today by Dr. Andy Bruner, who's an MDS specialist at our center. Hi, Andy.
Hi, thanks so much, Tim. So we're here today to talk a little bit about some of the new directions in MDS, and I understand that you've spent a lot of time looking at splicing factors in MDS. Could you tell us a little bit about those?
Sure. Yeah, several years ago, our group and several others reported that RNA splicing genes are recurrently mutated in patients with MDS. And there are a couple of features of these mutations that I think are particularly important for our discussion today. And the first is that they're common. So over 50% of MDS patients will have a mutation in one of these genes, making it by far the most frequently mutated group of genes that we know about in MDS. And the second is that they're acquired early. So we know that one mutation is not sufficient to cause MDS. Our cells have to accumulate several mutations. But again, a characteristic of these mutations and splicing factors is that they tend to be the first or one of the first mutations acquired in this disease.
So you found a mutation that we see in most patients with MDS that really seems to be early in the development of the disease. Why is that so important for our understanding of it?
I think particularly for thinking about developing new therapies, both of those features are important. First, again, it's common. With a majority of patients with MDS having these mutations, if you could find a way to target those mutations, you could have a big impact on the MDS population. And the second, the fact that they're early means that most, if not all, of the cells in the bone marrow harbor these mutations, as opposed to a later mutation that may only be in a few cells in the bone marrow. And we know from other examples that drugs that target these early mutations tend to be the most effective in oncology. And a great example is imatinib and other drugs that target the very earliest mutation in CML.
So if we can find some way to approach MDS cells that harbor this mutation, we may be able to better treat their disease. Can you give us any insight into how that might work?
Sure. The way we're thinking about this is trying to exploit a concept called synthetic lethality. And what that means is that when there's an abnormality, in this case a splicing factor mutation in a cell, it may uncover an Achilles heel, another pathway that cell is dependent on. So that if you target that pathway, the cell dies, whereas a normal cell might tolerate that inhibition. And again, a good example here in oncology of synthetic lethality is PARP inhibition in breast cancer patients with BRCA abnormalities.
So in MDS, it's somewhat like both the MDS cells and normal stem cells have both a regular generator and a backup generator. When you get a splicing factor mutation, you lose your regular generator, and then we think that somehow by targeting the other mutation, you're throwing a wrench in the last little backup that they have, and they stop working.
Exactly, that's the idea. So we know that the splicing factor mutations alter splicing, and this is a critical pathway, a process that's present in all of our cells. The idea is those cells are dependent on some remaining normal splicing activity, and there are now compounds that will target that. And we and others showed in the lab that just as you'd predict from the model, a cell that has a splicing factor mutation is much more sensitive to these splicing modulator drugs. And that kind of evidence provided the rationale to launch a clinical trial that's ongoing. And I know that you're directing this trial at our center, and it's probably too early to talk about results, but maybe you can share with us just the goals of the study.
Yeah, no, we're very excited. It's a phase 1 study that's looking at a drug that modulates the splicing factor, and it's first being tested just to make sure it's safe. But we're also looking to make sure that this drug is having signs of efficacy and perhaps improving transfusion requirements or delaying the transition into more aggressive forms of disease like leukemia. We're enrolling here, and there's sites across the country as well as around the world that are enrolling patients on this study. We've enrolled our first patient last year, and we're hopeful that in the next year or two we'll be able to see some really exciting results from this agent.
That's fantastic. So in parallel with that, continuing to work on these splicing mutations, we've actually in the lab uncovered another vulnerability, another Achilles heel in these cells. And there are drugs that target that pathway. So we're conducting experiments now in the lab, again, to demonstrate proof of concept that cells with a splicing factor mutation may be vulnerable to this other pathway. and then beginning to think about how we might be able to combine these two approaches. Since they work by different mechanisms, it's possible that these two drugs together might be more effective than either one alone.
That's really exciting. So we're essentially learning the core machinery of MDS cells and trying to exploit that understanding to develop new therapies. I'm really excited for developing some new drugs for our patients in the next years.
So am I. So Andy, thank you so much for taking the time, and thank you all for joining us for this update on MDS science from the MDS Foundation Center of Excellence here at the Mass General Cancer Center.